this group who never had immunology before or cell biology, where you would learn about these terms. So before the next lecture, before tomorrow, please read those terms and try to remember. If you absolutely know nothing about it, then try to look it up in more detail, because we will not have time to talk about that. But antigen can be anything that will initiate an immune response. It can be complex, or it can be a small antigenic determinant that is just one structure against which the immune system can be activated. So throughout the GI tract, we're going to see the lymphoid infiltrate, because antigens, that is everything we swallow, okay, can be antigen. Anything we swallow can initiate an immune response, and depending on whether we need to initiate immune response or we develop the tolerance against that material, will be sampled and basically thought about by the immune system. And the immune cells are everywhere throughout the GI tract, down here in the lamina propria, and these cells are constantly moving. They never stay in the same place. So whatever was here now, 2 minutes later, we're gonna be somewhere down here, who knows where, and they move in between the different parts of the secondary lymphoid organs, lymph nodes and blood and spleen. They circulate. Okay? So here we see the gastric mucosa. The surface is covered by simple columnar epithelial cells, which produce lots of mucus, especially in the cardiac region here. Down here in the glands, a number of regenerative cells, obviously at the bottom, and still mucus-secreting cells are present, and then we go to the muscularis layer. Even in the stomach, the muscular layer is inner circular, outer longitudinal, but it is not as well organized as the rest of the tubular GI tract. What else do we have to see? I'm going to go to the higher magnification, obviously, on this section, so you will be able to see these yeah, I just brightened. Oh, okay. That's the reason the condenser was down. So you see those very pink cells. That's because it's a past state, and they are stained pink. If I move back, this was the area where we're gonna meet here. You see the past positive. The deeper layer of the esophageal epithelium is also somewhat past positive, because these cells contain glycogen. Okay? We did not see the submucosal glands in the previous slide, so I'm looking here in the esophagus trying to find some. There we go. These are the submucosal glands. This is the muscularis mucosae here, and these are the submucosal glands in the esophagus, even close to the region where it opens into the stomach, but there is a submucosal gland. So remember, in the esophagus we see submucosal glands. In the stomach there are no submucosal glands. Okay? And the lymphoid infiltrate down here, again in the lamina propria. These are the gastric glands. And as we move further down, and we're gonna reach the main section, the corpus of the stomach, if we move far enough, then we will start seeing the other cell types in the gastric glands, such as parietal cells and chief cells. We are still not there yet, but we're gonna get there. There we go, here. Okay, here these gastric glands are going to be more straight, and you're going to see these straight, long gastric glands here, and recognize the parietal cells. I go to one higher magnification here, and just trying to get a little more light here. All right. There we go. You cannot miss these cells. These are these large, round nucleated cells are the parietal cells. Again, this is still the past staining. The cells in between them, the mucus secreting cells in between them, are stained pink. If we move down to the base of the gastric glands here, most of these cells are either regenerating cells or chief cells, and chief cells secrete pepsinogen. Okay? These are the chief cells down here in these glands. And then we see some blood vessels here, and the lamina propria infiltrated with lymphoid cells. We probably can see some plasma cells, but we already done that, right? There is a plasma cell right here,